So I prefer to start with the lipids and then go to the enzymes. Or they are same. There isn't actually priority for them. But you have enzymes here. So let's start with enzymes. And then you have lipids. So, sorry. And today, enzyme structure first session. We have like a first session. Yeah. By the way, I am Ehsan Khalili, or Ehsan Khalili, someone likes, but K is the correct pronunciation of that. I'm glad to see you. I will lecture with you. And so today and tomorrow we will have some sessions as usual. So we have to reorganize some of these schedules to include lipids and lipoproteins to our plan. And so today's session is a kind of thing for starting that I would prefer to start with one short movie, and you are familiar with that. Who can actually make a guess? Yeah, what is this? You can see the title. And over that title. Do we have sound? Yes.
Why is the fastest guy in? If you need something done, he's on.
I hope so. We are really fighting the clock with everything that counts. Wait. They're all slots? The clock in every minute.
Fastest guy in. If you need something done, he's on.
I hope so. We are really fighting the clock and every minute counts. Wait. They're all slots?
Are you saying because he's a slot, he can't be fast? I thought Zootopia anyone could be anything. Flash, 100-yard dash. Buddy, it's nice to see you.
Nice to see you, too.
Hey, Flash, I'd love you to meet my friend. Darwin, I've forgotten your name.
Officer J. Topsy, how
I am doing just
fine?
as well as I can be. What
Hang in there.
can I do
Well, I was hoping you could run a play.
for you
Well, I was hoping you could.
today?
Well, I was hoping you could run a play for us. We are in a really big hurry.
Sure. What's the plate
29 T.
number?
29 THD03.
Two, 9,
THD03.
T,
HD03.
H,
D03. 3.
D.
0, 3.
0.
3.
Hey, class, want to hear a joke?
No!
Sure.
What do you call a three-humped camel?
I don't know. What do you call a
3-pound camel.
3-humped camel?
Pregnant.
Yes, very funny, very funny. Could we please focus on the text?
Hey,
Wait.
Priscilla.
Oh, no! Yes? Flash?
What do
No!
you call
A 3-pound camel? Bring it! Okay, bring it!
Three.
Got it! Please,
humped? Here,
Yes, it is. Hurry,
You.
She's so pretty.
Go.
It's registered to Tetratown service. Oh, look, I took Robertson. Take a look at Tetratown. It's in Tetratown.
We're gonna hustle, bud. I love you. I owe you.
Hurry, we gotta get the rush hour in. It's night.
So, who wanna start the link between enzymes and structures? Why you are so lazy and so late? Their metabolism is low, and their enzymes and or subjects that we are going to study, they do everything so slowly. Instead of, let's say, I can produce or release ATP, many of ATP in a second to a lot of these things as you, and be so fast because of the enzymes. But their enzymes or their activity, metabolic activity is low, and mainly because of the enzymes, type of enzymes that they have. You can find enzymes everywhere. Let's start with some daily recommendation related to enzymes. In diets, fruits are everywhere that you know. Just give me some examples. You can use some information on this slide.
Like fruits. Say that stomach enzymes.
Stomach enzymes, exactly. Amylases, lipases. Just, yeah. You might have heard some of your parents or actually some seniors in your family, they say after a meal, take that fruit, specific fruit, they are going to say that it helps your digestive system to digest them or help you actually feel better after a heavy meal. So pineapples, kiwi, lemon. Mainly in kiwi, we have a specific digestive enzymes. That is the best suggestion for you if you are in a big ceremony and you are going to help your stomach to handle a lot of those things that you get. Get it just slice, 2 slices or 3 slices or whole of kiwi because it has a digestive enzymes. In Iran, kebab. You're familiar with that now. So one of the most favorite actually foods, and for preparing that meat, some people actually start to, marinating that or adding some seasoning that. But some people, they don't have enough time. They just add kiwi, but they start just 1 hour, 13 minutes before, doing kebab. Because if you add kiwi the night before that, it can digest just amino acids. You will not have a meat structure. So it has a great digestive, or pineapple is another best digestive sources of enzyme. And in some medication they are used. You're going to see some drugs in the pharmacy, their sources are from these fruits. Or in detergent. You might have heard some of this detergent, they have enzymes to make everything so clean. And some of these enzymes in pharmaceutical industries. You can find some of them. Some people may have a issue with digesting many of foods, and they get some of these enzymes as a drug. So, see the importance of them. You can find everywhere. In clinical application, you are going to see that one of the best tests and most important tests for emergency unit for assessing myocardial infarction is enzyme. What enzymes? We have a specific enzyme like troponin or some proteins or AST. In clinics, we use some of them. You are not going to study this here. You will study this thing. This is just example for you. We can use them in clinics. If you go to hospital, in lab, you will learn some of these enzymes are getting increased in blood circulation after some toxicity events to your liver or your muscles or your heart muscle, and they increase in blood circulation and you measure them. I'm not going to explain all of those details in the slides because I'm explaining them, and I will not read them word by word. Or in cosmetic. Many people actually use this kind of science to get rid of some of those diseases they have. Might be because of the accumulation of some lipids in their skin, and they use some of this. However, I'm not recommending that. It is better you do it based on the science facts. Who is interested in making jelly? Have you ever tried making a jelly and putting some of this, pineapples on it or some kiwi on it? Because that is so, it seems so tasty when you see a jelly that is clear and you see some kiwi part or pineapple inside that. If you try it, you might not see your jelly works properly. Why? Because that enzyme in pineapple and kiwi digest the jelly. What are the contents of the jellies? Are proteins. And they have digestive enzymes. They have proteases. They digest that. What is the solution for that? You put your pineapples or kiwi one night in freezer and kill enzymes, and then you can put it. Because that enzymes are dead already. And so that's just cooking recommendation. Keep prepared. Every time you will see enzymes everywhere. If you feel full after a diet, it is better to get a pineapple or just kiwi or lemon. It is the scientific base of how this pineapple or kiwi digest the jelly, because they have proteases, and we know if you digest that proteins, that structure will destroy. And this is the reason for that. You can read that. So all things that you are going to study is like this. How interested are you? Very good. MashaAllah. Yeah, it's great. Best thing that we can say from creation or everyone. SubhanAllah. Yeah, exactly. Yeah. Every time that I look at a lot of these things, they are working and without making any trouble to others. See, a lot of these enzymes, all of these reactions happens in one cell, and all of cells. In some cells, some of these pathways are like highway pathway, they are more dominant, but the others are also there. So, but you are not going to learn. Luckily, you are not going to learn these things. Or, fortunately, there is a just 6 ways to doing all of these reactions. Can you believe that? You are going to just learn that 6. See, I shortlisted all of this enzyme in just 6 groups. You are going to learn that. That's so easy. But that's a clever idea. See, all of these reactions are done just with 6 type of actions. And that 6 type of actions you will see everywhere in reference. So, this is the learning objective for this session. You will learn what are the enzymes, how they work, and what environmental factors affect them, and classify them, like how many are there, and then the best usage of that. How much drugs are essential for control? You study these enzymes. If you look at drugs as a doctor or pharmacist or everyone that is involved in healthcare system, you are going to see many of the drugs that you prescribe, many of them target enzymes. And you will learn how to use these drugs or how these drugs target that enzymes. Or we call it, kind of inhibition of this enzyme. You will see that. Why we need to study the behavior? Because we need to design drugs or give them enough concentration of drugs. How much of one, let's say, when you take a painkiller, how much of painkiller do you need? 1 milligram, 2 milligram, 10 milligram, 1 kilogram, how much of that?
1,000?
Yeah, you are good. You can give yourself. Right now for yourself. Yeah. How much?
100?
We don't know. It depends on the enzymes and level of the substrate that inhibits that. Sometimes if you get more of these drugs, what happens like acetaminophen or paracetamol? What happens in a low level of that, it is good. But in high level, it is toxic. Even your carbohydrate, when you need that, the level of that you need that is so sweet and good. But if you consume more of that, what happens? Diabetes, obesity, a lot of things. See, even there is a wise sentence. It says, try to be your drug, your foods be your drugs. You should get them. Next time you are going to get carbohydrates, let's see how many of carbohydrates I'm taking. So this level is good, and this level is toxic. Yeah, what are the enzymes? Can you define them? Do you have any idea? Based on their structure, they are protein, carbohydrate, lipids. What kind of them? They are. However, they might have some carbohydrate in their structure, but mainly the main structure is protein, and the function part is protein part. And what does they do? Exactly. They convert something to other things. Sometimes they break down, they making larger things, a lot of things they do. Those 6 groups that you can see, they just do 6 types of the work. Are they used or altered during the reaction? No. Yes. They are not changing during the, not changing. They have some small changes, but based on the vitamins that they have. Mainly for enzymes, we have a vitamin part. That vitamin part are changed, and the enzyme part is not changing. And another important thing is they are reusable. They are reused again in another reaction. They can actually do this reaction and then does a lot of time. So that's the definition of it. So, what parts you can find in enzyme structure? Protein parts and non-protein parts. See, this is a protein part, this is non-protein. This is an enzyme or holoenzyme. We call it holo means total or whole something and complete something. So this is a holoenzyme, and it has 2 parts, protein parts and non-protein parts. The protein parts is called enzyme. You might see just some places, but mainly they are called enzyme. But we are gonna, focus on some details here because these details for a starting point is important. We have enzyme and non-enzyme. protein part. These non-protein parts are called cofactors. Cofactors, or if you look at the name, you can find the meaning of it. Cofactors. They are which enzymes they help for enzyme for working. These cofactors mainly, most of the time, they are vitamins. Because of that, vitamins are essential. Without these cofactor parts, it seems we don't have this enzyme. Can you give me some diseases related to enzyme insufficiency or deficiency?
Lactose intolerance.
Lactose? Anemia? Edema? Sorry, I didn't get that. Edema? Edema? What's the edema? What's the insufficiency of that?
Water is stored in excessive.
Yeah, actually, you mean edema? Yeah, edema is a general, actually, condition. It can be because of the several reasons, but specific diseases like albinism. Do you remember some people, they don't have color on their eyes, or they actually, that's it, just for instance, one example. It doesn't go properly. Or some congenital diseases related specifically to Huntington, yeah, specific protein. Yeah, it's not enzyme here, but that is a protein. All proteins are not enzymes, but all enzymes are protein. Yeah. And many diseases comes from enzyme insufficiency. Even when you get more compound, you can inhibit some enzyme to start another pathway, like glucose. If you get more glucose, what happens? When you have enough glucose, it goes to the synthesis of the lipids and inhibits some pathways, even that glucose. So, these coenzyme parts mainly are vitamins, and in some cases we have ion, like metal ions, like copper, iron, zinc. A lot of those metals, they can be used as a cofactor. Those enzymes are called metalloenzymes. That is so. Metalloenzymes. They have metal ions, and their structure for work. So you can see the cofactors here. I'm going to explain it more. Other things about the enzymes. In their structure, they catalyze a reaction. Let's say, for example, this is a substrate, I'm an enzyme, and I'm supposed to transfer this one from this to other one. Doesn't go here. I'm just trying to show this one. This one is not that. Let's see here. Here. Yeah. I transfer this one, this part, this red cap, from this one to the other one. This is a transferase. This is the enzyme called transfer. You can see that. But for doing this, for catalyzing this, I need a specific active site. Part of enzymes, let's say these are my active sites for doing this. If my active sites are occupied, let's say I have another thing. Can I do this? No. Because my active site is important for doing this. All enzymes, they have a specific site for catalyzing the reaction. We call it active site. This is the active site. So, what are the properties of active sites? Active sites are specific for example, for this substrate, let's say here. And see, we have a substrate here, we break down, we just break down, remove that thing. But, what is the shape of this active site? We have two models. What do you think about these two models? Let's say, first one is like key, lock and key. Let's say my hand is like this, it comes here and goes like this. It's a good idea, lock and key. In lock and key model, my hand is just specific for this one. How about, let's say, we have some other things here. 14, let's say. Can I actually catalyze reaction for this one? Then actually my active site is like this. No. What model do you actually become in this big model? My active site can actually change its shape for this structure. So these are two models for the enzyme active site structure. Some of them lock and key, some others. It's a kind of giving specificity and what is the logic behind it? What do you think? Why should we have some enzymes lock and key, some others induced key?
For the places. For the places in the body.
For
the places. What do you do?
yeah, for specificity, mainly for specificity, this is more specific for this, and more publicity. Because it gives that option, it can move in a range of components. Instead of that, our body had to produce a lot of enzymes. For every compound, we should have a specific enzyme. But here, some enzyme can work on a group of things. Let's see some examples. Here. This is a lock and key model. You see some information of them here, I explained them. In lock and key, like this lock and key we have here, the active site is just specific for that substrate. The other substrate can't actually go there or be catalyzed. Sorry, I should actually do this typing sounds here. See, in active sites, this is the lock and key, this is the active site, this substrate is specific for this area, and this enzyme catalyzes reaction. This information also for you, when you review that one, you might need to find, I'm not attached to these slides, or my voices are not attached, and some of those information is for you when you review the slide. I'm not reading those sentences here, I explained it later.
Tell us about which one is for examination.
I will give you some template questions. You can actually find what parts are important. I will give you some template from previous semester's questions.
You mean transmitter?
Yeah. You can see some of those questions are actually those types of question or which part are important for you to study that things. Yeah. Yeah, for the last year. Do you have them? You have collected them. So if you have them, so if you have it, that's good. So you know how which parts are important and what's the main idea for it.
But we still need of that content.
I will send you.
Okay.
So there will be no new ideas, except for the past papers.
I always give those questions. If I actually design a new question, what happens? It seems for every class I should design several times of the question. It's not fair. I should check. For 3 years I wasn't in Iran. For 3 years, 2.5. Some of them are not mine. I'm not sure, but I will send you my own question. Yeah. So, about the induced heat model, the simple we have an active site like this and a substrate like this. They are not same to each other, but our active sites change some structure or rearrange structures of active site, and it is fit to this substrate. Let's go some examples of it. Why we have this one? For specificity, mainly it's for specificity. So our enzymes, some enzymes are specifically for a specific compound. Like example, urease. You can find the information on it. What is the urease? Acts on just urine.
ALP.
Or glucokinase. Acts on just glucose. The name says this enzyme works on just glucose. How about the hexokinase? What do you think? Six carbon sugars, no matter that is a glucose or fructose, galactose, all of those 6 carbons. But the second one, what's the second one? Second one is a group similar substance. The first one, urease, was a specific. It's just a single substrate, or glucokinase. Every time that you want to study something, try to find the example. Without the example, it says you haven't learned it completely. Here, so you can write examples. For this one, urease, or glucokinase, or everything that you find that is a specific for that compound. A group of similar, like hexokinase, the best example compared with the glucokinase. How about the particular types of one? You can give me an example, you know. Peptidase, or lipase, or amylase. See, they are enzymes, but they work on a specific bounds. Those three examples are working on what kind of compounds? Carbohydrates, lipids, and proteins. See, the creation is so clever. It says, I have some enzyme, they just act on a specific compound. I have some enzyme, they actually cover more. And I have some other enzyme, they cover more that we can actually. They cover carbohydrates, they cover peptides, they cover proteins. So the enzyme protein is like that in our body, and they are classified in these 3 groups. You can find the previous slide examples also here. This is a chymotrypsin. What is this chymotrypsin? It's a digestive enzyme. It's a peptidase, generally. For hexokinase, you can see here for urease, here also glucokinase, the first. So, how does work this enzyme? The decrease energy of activation. Imagine without the enzyme, what happens? Let's say without me. What happens to this one if it's supposed to break down?
Like a slope, Zootopia.
Yeah, exactly. Thank you so much. So here, when this one is supposed to remove or transfer to this, what happens? It takes million years to remove that one without the enzyme. Or you can do it with a lot of energy. You can put on it how, I don't know, burn it or adding in a specific area to this everything I remove that one. It needs a lot of energy, but in the biological system, it's not applicable. We can't actually have that temperature for doing this reaction. What's the best solution? Enzyme just decrease that level of the activation. For doing all of these things, we need to activate that, add more energy to that one, but in some cases it's not applicable. The best option is enzyme. Actually, they reduce that activation energy with some like here. Some enzyme, some parts of the enzyme comes here to but that one and just remove without putting a lot of energy. However, in just one group, we need use ATP for reaction. The others actually do the reaction without ATP. Just one group of them just consume ATP. The others, they are. I'm going to explain that. I'm just mentioning that for doing that enzyme, for reducing activation of energy, they don't use ATP. Just one actually. One of them actually.
If you have extra enzymes, you still have, right?
Yeah. So,
All the food actually from science plus one to my have
so we're not I don't like that one. Just in 10 minutes, I will give you a rest and then rest. But today's session, after this session, we will have a practical session. We will have a practical session. Yeah. It helps you actually wake up. So, no, just after 7. Yeah, I know. This time of the morning, having enzymes, biochemistry, at the weekend, yeah. But I hope you find some practical usage of these enzymes. And yeah. So it's like that. For doing the reaction, it needs a lot of energy. Without the enzyme, it seems, not someone actually who does that, but it seems it needs a lot of energy to do that reaction. But with enzyme, it seems it doesn't need a lot of energy to doing that reaction. So in just one sentence, what does the enzyme do? They reduce activation energy for reaction. And this one, we have some rules for classification or naming of enzymes. Some of them, classified one, we have some classification for them. I'm not going to learn them, just learn the general naming. For the enzymes, they end in what? A's. How about the sugar? They are ended in O's, exactly. So you can actually make a guess about the specific enzyme for a specific substance. For sucrose, what is the enzyme? Sucrose. And for amylose, amylase. And so on. You have sounds like this. Some of these enzymes, sorry guys. Sorry. Yeah. And many of these enzymes actually gives you enough information about the reaction they do. Like oxidase, they oxidate, or reductase, they reduce something. Or transferase, they just transfer. Or epimerases, they just convert something to other same structure. Yeah. And some of them, they have an ancient name, and so when they actually discover, they actually get some names for the reaction that they does, but after that, so we had a lot of enzyme, and science is actually was based with a lot of names, and they had to have a specific classification for that. So the classification is like this. We have 6 groups. All of those reactions that you saw in the first image about the enzyme, they are catalyzed by these 6 groups. First group, oxidoreductase. They oxidize or reduce something. The second group, transfer something. The third group, is hydrolyze, add water. Have you seen some of those movies? They use water for breaking down everything. Our body does that before. So the best engineer are inside. Add water, break down everything. So yeah, we already have it. And the other groups are liases. I'm going to explain that. They are same to liases, but in some other way. And this one, isomerase. It's the isomer of something. Yeah. And finally, ligases. Join, add something to each other. The only group that use ATP is this one. Ligases. So you can have, yeah, 10 minutes is good. Go and get coffee. Yeah. 15 minutes. It's good, 15 minutes. What are you worried about?
We have to complete this. I do we need enough time to complete which topics are we going to complete today? Enzymes and lipids?
Yeah, I will submit this today.
Today?
Yeah, I will submit. I have to come back though. I have to come back. Yeah, I have to see time. Yeah, to cover that. This enzyme, oxygen reductase, you have different types of oxygen reductase. See just some names of them because in many places of the book you might read about the enzyme. You just need the general names of them. Here we have a compound like lactate and NAD. I will talk about this NAD, but that's a kind of like B3 derivatives. I will actually explain those vitamins. But yeah, here we have covered lactate in the body to hydrate. One of the most common actually examples you will hear a lot about the producing lactate. You might say, I did some lot of activity, I now produce a lot of lactate in my body, so this is the reaction is like this. But we produce lactate and pyruvate convert to each other like this. We have an enzyme here that is called lactate dehydrogenase. So what's the dehydrogenase? It's a kind of oxidoreductase. In oxidoreductase, you see some examples here. For example, oxidases, peroxidases. What is the peroxidase? Yeah, hydrogen peroxide. So we use this hydrogen peroxide for what? For killing bacteria by neutrophils. See, we have a lot of this thing. Or oxidation of lipids, or many of things. All of these enzymes are essential. Without one of them, we will face with a severe disease. Because they are group of the enzyme they catalyze this reaction. And what kind of enzyme activities are, kind of screening for new births in your country? Do you have a list? Are you familiar with some of those genetic tests after birth, some screening tests? Yeah, for thyroid function is one of them. For newborns. And for phenylketonuria. Phenylketonuria is specific for enzyme that's called phenylalanine hydroxylase. It's related to this group of the oxidase. See, they are important. You can't actually ignore them or underestimate their importance. Everything in disease is related. Everything in drug prescription is related to enzymes. And see, so you can find different types of oxidoreductase. You are not supposed to memorize them. You can just make a guess of that. Here, everywhere that you see adding oxygen, removing oxygen is a kind of oxidoreductase. And the other things that is important, adding hydrogen or remove it. See, we remove transfer here, we transfer, that's not transferase because it's different. We remove 2 hydrogen from this structure and convert it to this one. See, This is an oxidoreductase, even hydrogens. You might say, is it a transferase because it transferred? Hydrogen transferring isn't transferase. It's oxidation or reduction. Just in transferases, please look at that one. You might say here, yeah, you transfer the hydrogen from this one to this one. Is it transferase? We are not calling that one. Transferases, except this, yeah, hydrogen. That's the other groups. We have amine groups here, transfer to this one. What happens? This one transfer this amine group to this one. We produce what? Glutamate and pyruvate. From what? Alanine and ketoglutamate. The details here isn't important. You are not going to actually say, I will just ask you, one of your questions is going to be one of these examples. So what type of the reaction? Even I'm not asking about the name of the enzyme. I will just ask a group of that 6 group. Which group is that? See, here we transfer this amines to this structure. What is the importance of this enzyme? What do you do? We produce what? What is that? Glutamate.
It's a precursor to lipolysis. Energy.
We produce non-essential amino acids. Why some of amino acids are non-essential? Because we have this enzyme. Without this, the rest of the non-essentials could be essential. So we can synthesize half of the amino acids in body by this enzyme. Great one. So this is a transferase. Like transferring this group to this one. Sometimes exchanging between them. This is a transfer. So the other one, example. See, we added water to this structure and converted it to compound. Like, let's say we have a lot of units, local units. See? See? Maybe you should imagine. So, we have units of the glucose in starch or in every carbohydrate. Hydrolysis add the water to this part and remove. Oh my God. Glucose. We remove these units. Hydrolyses are used to remove one unit of something. Here we had 2 of these units. Why I just emphasizing on units? Because the next one is a confusing one, lyases. In lyases, if you look at the structure, the definition, what it says, if add water, you might say add water. In previous one, we add water and remove that unit. Are they same? No. In some cases, you can add water or remove the water, but you can change or rearrange that. It's not isomerase. It's rearranging of something. The most complicated one is lyases, because more than one kind of reaction happens in. Sometimes add water, sometimes remove water from that structure, sometimes make double bonds. It's called lyases. Again, you will see some examples here. See, this adding water, did we remove unit here?
No.
No. In hydrolysis, when you add water, what it says? Remove units. Remove units, not just one part. Unit. Glucose. Amino acid. You remove amino acid. So the differences is there. Do you have any question here?
How could we identify it from, like if we are doing a reaction, it's not like this, it's a different type of reaction. How will we identify lysis from hydrolysis?
So in hydrolysis, all the time, if you add the water, you will see units are removed. But in lysis, it's not like that. You are not removing units. You are just rearranging that shape. Here we have an extra units here, even reverse reaction. Let's say this reverse reaction happens. Here we added water. Imagine we remove water here. Remove the water from the old water we have here. Rearranging this. See, this compounds with double bonds. Sometimes removing water makes double bond. It's called lysis. So the big difference between lysis and hydrolysis, in lysis we add water to break it down units, same units. But in lysis we could have actually water or remove that one, but the structure has just one more molecule, one molecule has a change in it.
So it's an addition reaction.
Just focus on units. In hydrolysis, when you add breakdown in units.
Okay.
Otherwise, that's lysis. If you see adding water or removing water. Isomers, they have stories. It seems you have it like this and taking you out. This is isomers. Examples? The carbohydrate? Fructose and glucose, lactose and glucose. They are L and B, different types of isomers. L and B, like here. Glucose and fructose. And like this, the last one. Then you have a molecule like this, and you are going to add something, not transferring. Add something, extra something, this structure, and you need to make some fusion on it. You need energy because carbon, nitrogen, carbon bonds need more ATP, energy for making that bond. The last group is like that. See, we use ATP to synthesize new larger molecules. See, this one, this amine groups. See, this amine groups is here, and we use this ATP as an energy for this. The energy are stored here. If you break this bond, break down this bond, that energy released. What is like that? Everywhere that you save the energy for making a bond, next time you are going to break that bond, it releases energy. In the nature is like that. What are the sources of the energy in the nature for our body? We get energy from what compounds? Carbon, glucose, mainly, or lipids. So they have carbon bonds. And bodies actually, what does do? Just breaking down those things in metabolism, you will learn that. Breaking down, and in carbon we have energy. The magic is here. We have a system, burn those things, carbons, and extracting energy. It is our magic. And releasing CO2 to the nature. What is the next part of that? How actually those CO2 in the nature are converted again to carbon. Plants. How they actually make a bond between carbon. Where does come from that energy? Sun. Photons. They receive that energy. I was always thinking, if we had a chlorophyll, we could have it. We don't have to try to get carbohydrate from the out sources. We can synthesize them, and we were, effortless for getting all of this carbon from outside. There is a, great stream in the nature for producing, consuming that, releasing it in the nature. In Princeton University, 2 years ago, they tried to condense, one of the solutions, what is that for the high CO2 in the temperature, condensing them. We know that actually jungles are essential for that, for capturing that CO2. But we know we have a deforestation and we don't have enough jungles to actually condense them. Another solution is that condensing or putting them in the oceans. Another one, in some university, they are thinking they actually do the condensation in the lab. Wow. The same happened in the Middle East and some parts of the world for a million years. Condensing carbons and producing what? Oil. Yeah. In university, they condensed that. They connected 6 carbons to each other. And they are going to produce oils or gasoline or petrol from those. But putting together carbon-carbon needs more energy. And so that energy comes from what sources that is in. But we extract that energy from the water. So why I explain this one so much? Because when you are going to make more energetic bonds, you need to put energy on it. But you are not wasting energy on that part, because body knows how to extract or saving that energy on that bond in the next step, in some other place, and extract that energy. Also, you will see these tables, they are actually kind of overview of those things that I explained. They are same actually explanation that I gave you, also this one. I try to put some different types of the explanation because one explanation isn't good for everyone. Sometimes some guys are not happy with just kind of explanation. In another explanation, they might get that concept so easy. They are same. They are not different things. This 3%. So you can open the other one.